Welcome my colleagues and my dear students, and today we are completing the story of transport through the cell membrane. As we said in the previous lectures, we have three methods for transport through cell membrane. First, diffusion; second, active transport; third, the vesicular transport. The first method, which is the diffusion, occurs everywhere in the universe and it does not need energy. However, active transport is a vital process that occurs only in living cells, where substances are transported from low concentration towards high concentration, and this necessitates the presence of energy. So, what is the definition of the active transport and what are the main differences between it and the other methods of cell membrane transport? Active transport means transporting substances across the cell membrane from areas of low concentration for this substance towards area of high concentration, and there must be presence or consumption of energy in the form of ATP. We have two main types of active transport, whether primary active transport or secondary active transport. The difference in the methods of use of ATP. If ATP is consumed directly over the carrier by the ATPase that is present as a part of the carrier protein, this is called primary active transport. However, if the consumption of ATP is indirect, so that the carrier has not ATPase and the ATP is used elsewhere in the cell, as a result of this step, the process is called secondary active transport, where consumption of energy comes by indirect manner. So we have primary active transport and secondary active transport, and we are starting now to discuss primary active transport. We have the general idea that it needs the energy, that it needs the carrier, that it transmits substances from low concentration towards high concentration. Note this picture. You transmit substances uphill from low concentration towards high concentration. We have 5 important characters for active transport, whether primary or secondary. First, it occurs against the electrochemical gradient, as we said, from low concentration towards high concentration. This, of course, serves a certain biological need. Second, it is active. It needs energy in the form of ATP. Third, there must be carrier, whether in primary or secondary active transport. And when we mention carrier, we must mention 2 important characters. As we said before, with facilitated diffusion, there must be rate limitation and there must be saturation. So the process of active transport occurs in living cells. It needs ATP, it needs carriers, and it is rate limited and saturable. We have 3 main examples for the primary active transport. The most famous of them is sodium-potassium pump, sometimes referred as sodium-potassium ATPase or even sodium pump. Then calcium ATPase or calcium pump, hydrogen-potassium ATPase or hydrogen-potassium pump. Calcium ATPase is present in the wall of the sarcoplasmic reticulum in muscle cells. The second is hydrogen-potassium pump, and it is present in 2 sites inside our body. It is present in the stomach and in the kidney. Sodium-potassium pump in detail. What is the pump? What is its importance? What about its structure and how it operates? First, the sodium-potassium pump is widely distributed inside our body. It is present in the wall of all living cells. About 40% of the energy consumed inside our body is consumed by sodium-potassium pumps, which are present in millions in the cell membranes of all living cells. Regarding its structure, it is formed of 2 subunit. First is the alpha subunit, the second is the beta. We have 2 alpha and 2 beta subunit. Beta subunit, its importance is simple, just fixation of the pump in the cell membrane. However, most of the vital functions of the pump is done via the alpha subunit. This is the inside of the cell. This is one of the body cells, and this is the outside, which is the extracellular fluid. First, look at the concentration of sodium and potassium. The concentration of sodium inside the cell is 14 milliequivalent per liter. However, outside it is 140. So sodium is 10 times higher in concentration outside than inside of the cell. Regarding potassium, it is present extensively inside the cell. Its concentration inside the cell is 140 milliequivalent per liter. However, it is present in a low concentration in the extracellular fluid, only 4. So it is present 35 times higher inside than outside. This is a rule in biology, is that sodium is present mainly outside the cells. Potassium is present mainly inside the cell. This is the general rule which is responsible for regulating vital functions in biology. So how these concentrations are made and how they are maintained? The secret is the sodium-potassium pump, which is present in millions in all cell membranes, and it is composed of the following components. First, 3 binding sites for sodium ion that are present towards the inside of the cell. They are present on the alpha subunit, as I said. It works to pump sodium from inside to outside, so logically the binding site of sodium must be present towards the inside. Second, the 2 binding sites for potassium which are present outside. It works to pump potassium from outside to inside, so logically the sites for binding are present toward the outside. Then it has ATPase binding site which bind a molecule of ATP and it produces energy. Then it has pain binding site, a regulating site which when bind to a pain or similar drugs, it works to regulate the pump activity, which is helpful in treating cardiac muscle abnormality. This is the inside of the cell. This is the extracellular fluid. You can note easily that sodium is concentrated outside the cell and its concentration is low inside. The difference is 10 times higher outside than the inside, and potassium is to some extent the opposite. However, the ratio is higher, where potassium inside the cell to the potassium outside the cell is about 35 times. These essential distributions are the main determinants of the biological life. You must know it by heart. This is a simple figure or a simple animation that describes the activity of sodium-potassium pump. As you see, it works for life. However, the degree of activity may increase or decrease. First, it binds to the 3 sodium. Then the alpha subunit becomes phosphorylated. Third, it releases sodium. Fourth, it binds potassium. Then it dephosphorylates. Then it releases potassium. To summarize these steps, we have 6 steps in the operation of the pump as described in the previous picture. First, the 3 sodium bind to their side towards the inside of the cell. Then, following this step, the ATPase becomes activated and it picks up a molecule of ATP and converts it into ADP, and it phosphorylates the alpha subunit. Once the alpha subunit is phosphorylated, it is now activated and there is a change in the shape of the pump, causing a release of the 3 sodium outside the cell. These first 3 steps belong to sodium ion. We have another 3 steps belong to potassium ion. The 4th steps, or the first of the 3rd steps regarding potassium, is that potassium binds to its sites on the outer aspect of the cell. Then the alpha subunit becomes dephosphorylated. This dephosphorylation induces another conformational change, a change in the shape, which causes release of potassium to the inside of the cell. What about the control of the pump? This is important, and this is clinically applied in many important situations. What are the activators and what are the inhibitors? The first and the second, they are logic. The activators are high intracellular sodium and high extracellular potassium. High levels of sodium inside or higher levels of potassium outside, they urge the cell pumps to work in order to get rid of the excess sodium inside and to get rid of the excess potassium outside in the plasma, which may be deleterious to many organs, especially the heart. So we can expect easily that high intracellular sodium, high extracellular potassium are activators, and the opposite is true. Low intracellular sodium and low extracellular potassium inhibitory to the pump activity. Availability of ATP activates. However, presence of little ATP decreases the pump activity. The pump is controlled by many hormones and chemical substances, and this is the base of its clinical application. Hormones like thyroxine, insulin, and aldosterone, they are activators for the pump. However, materials like dopamine, wapine, and digitalis are inhibitors of the pump. We finished the primary active transport. What about secondary active transport? It is a very important mechanism in biology, and it is responsible for many transport processes through cell membranes everywhere inside our body, and it is dependent on primary active transport, so that there is no secondary active transport without primary active transport. Secondary active transport also is transport of substances from low concentration towards high concentrations, and it consumes energy by indirect manner, and it is dependent on primary active transport. So what is the idea? Let us remember important facts. First is that the sodium is present outside the cell by a concentration of 140 milliequivalent per liter, and it is about 14 milliequivalent per liter inside the cell. Usually the force that urge ions to move from one side to another is one of two forces. Either the concentration gradient force, which pushes ions to move from areas of high towards low concentration, or the electrical force, where positively charged ions tend to move toward the negative areas of potential. So while you are thinking of the movement of any ion, you must search for two important points: its concentration and its charge on both sides of the membrane. Applying these ideas to sodium, we find that it tends to move inside the cell because its concentration is higher outside than inside by 10 times. Second, it is positively charged while the inside of the cell is negatively charged. So all the time sodium tends to enter the cell when it finds a way, and this is the secret for the transport processes through the cell membrane and the generation of electricity through the cell membrane. In the secondary active transport, we have principles. The first of them is that sodium is transported from outside to the inside of the cell. This direction can't be changed from outside to inside and from high towards low concentration. And there is accompanying substances that are transported together with sodium. These substances or solutes, they are transported from low to high concentration, whether in the direction of sodium from outside to inside, or even in the reverse direction from inside to the outside. We have two important examples for secondary active transport. First, the sodium glucose co-transporter. Second, sodium glucose antiport. This co-transporter for sodium and glucose. When we say co-transporter, This means that it transports the two substances in the same direction, so that you can guess that sodium and glucose enter the cell in the same direction. So what about the mechanism? This is the lumen, which may be the lumen of the intestine or the lumen of the kidney. You can note well that the concentration of sodium outside is high and inside is low. The other substance which needs to be transmitted, the glucose, inside the lumen its concentration is low. However, its concentration inside is high, and we need to transmit it, the glucose, from outside where its concentration is low to the inside where its concentration is high. Then it completes its path to the inside of the body. First, sodium and glucose bind to the specific sites on the carrier protein. This carrier protein is designed, is programmed, so it works only when it binds sodium and glucose. If one of them is bound only, it can't operate. When they bind together, it operates and it transmits them to the inside of the cell. You can note well that sodium is transmitted from high towards low, from outside to the inside. However, the accompanying substance, which is the glucose, moves from low towards high concentration, from outside to the inside in the same direction of sodium, so the process is called sodium glucose cotransport. The second example is the sodium calcium exchanger. It is present in the ventricular muscle cells, cardiomyocytes. Suppose that this is the cardiac muscle cell and this is the extracellular fluid. As you note, sodium is concentrated outside, further more than the concentration inside. This is our general rule. However, calcium is present in excess outside more than the inside. And for the stability of the cell function, it has to maintain very low calcium ions, because calcium, even if it is so little, it disturbs the cell contractility, so it has to get rid it of, and it is not allowed to appear inside the cell except during the process of contraction. So as you note here, sodium binds to the carrier from the outside of the cell. At the same time, calcium binds to the same carrier from the direction of the inside. The carrier is programmed so that when they bind sodium from outside, calcium from inside, it operates and it moves the ions, sodium from outside to inside, from high to low, and the other ion, which is calcium, move from inside to the outside, from low concentration towards high concentration. Finally, we reached the end of this lecture. See you again and goodbye.